Hi, everybody, and welcome to this morning's webinar talk hosted by Precisionary Instruments. I'm Abby, and I wanted to introduce you to Professor Mingfu Wu, who today is going to talk to us about his work in cardiomyocytes. So Dr. Wu is an Associate Professor of Pharmacology at the University of Houston. He has a background in genetics, developmental biology, and cellular biology. And his research focuses on unraveling the intricate mechanisms underlying congenital heart defects and left ventricular noncompaction. So his lab uses genetic, developmental, and cellular tools to investigate the etiologies of cardiac malformations and explore potential therapeutic interventions. And in this morning's webinar, Dr. Wu is going to dive into the fascinating world of cellular organization, or morphogenesis, in ventricular wall morphogenesis, shedding light on how disruptions in cellular organization can lead to congenital heart defects. So with that, Dr. Wu, I want to hand it over to you. And everybody, please feel free to ask questions in the chat box.
Great. Thank you, Abby, for the introduction. It is really a great pleasure and honor to be invited by Precisionary Instrument to introduce some of the projects that the lab is doing. So today we're going to focus on the cellular organization in ventricular wall morphogenesis. Okay, so I'm going to introduce ventricular wall morphogenesis, and then we're going to elucidate the cellular mechanism of a trabecular morphogenesis. Trabecular morphogenesis is one of the key steps in the ventricular wall morphogenesis. And then the third, we're going to talk about the beta-1 integrin, regular cellular behavior and the cardiomyocytes organization during ventricular wall formation. Okay, so the cardiac morphogenesis is a very complex process. And to not complicate, to not confuse you, I'm going to use a very short introduction about this morphogenesis process. So majority of the cells in the heart are derived from the two pools of progenitor cells, called the first heart field progenitor cells in red here, and the second heart field progenitor cells in green here. So those progenitor cells will migrate to the midline to form a heart tube. This heart tube will undergo S-shaping morphogenesis to form this S-shaped heart tube. And this heart tube will undergo further morphogenesis to become a full-chambered heart. So what I want to tell you is this is a very complicated process. And what we're going to focus on today is only the heart wall morphogenesis. But even the heart wall morphogenesis is very complicated. I arbitrarily divide this process to three steps. So the first steps are the single cell myocardial layer to multiple cell thick layer. So here's a heart tube at the bottom, embryonic day 7.5 or E7.5 in the mouse heart. So the heart tube contains two layers of cells, the endocardial layer and the myocardial layer, and they were separated by cardiac jelly. And so in this first step, the cardiomyocytes has to be multiplied to form multiple cell thick layer, myocardial layer. And in the second step, it is form this protrusion here. So this, as you can see at about E9.5, you can see this protrusion. This protrusion we call it trabecular. So the formation of a trabecular, we call it trabeculation. So the trabecular cardiomyocytes in red here are different from the cardiomyocytes in the compact zone in yellow here, as far as expression profile, proliferation, and also maturation status. They are quite different based on the single cell sequencing. And what I want to tell you here is the endocardial cells and the cardiomyocyte interaction here is very critical for the trabecular morphogenesis here. We know the signal pathways such as NOTCH1, unraveling, VEGF, cephalosporin, and angiopoietin, and also many other signal molecules are involved in this process. And the signal interaction here is so critical. That is, if disrupt this signal interaction, the trabecular formation defect will cause the trabeculation will fail to form, and it will cause embolic lethality. And one of the project, the key project in the lab is a signal interaction between the endocardium and the cardiomyocytes. And we have a manuscript submit to science on the revision now, and we hope we can get this done soon. And the third step for ventricular wall morphogenesis is called ventricular compaction. So as I told you, there's a trabecular here, and this trabecular eventually will correlate with compaction here. As you can see, the yellow cells are the compact cardiomyocytes, while the red cells are the cardiomyocytes from the trabecular zone. So this compaction will thicken the compact zone to make the heart beating stronger. And the significance of this study is the abnormal compaction will cause left ventricular non-compaction. And this is one of the major focus of the lab. That is how the LVNC is formed and how we can treat the LVNC. Okay, so let's move to the second one. That is elucidate cellular mechanism of trabeculation, trabecular morphogenesis in the mouse. Again, it's going to be very fast because it's already published. So here's our hypothesis. We hypothesize that a direct cell migration and oriented cell division contribute to the trabecular initiation and formation. So again, here's the endocardial cells and here's the cardiomyocytes. So the cardiomyocytes, they can display all perpendicular orientation and only the direction migration to the cardiac jelly to form trabecular. And also the cells can only go perpendicular division and send the daughter cells into the cardiac jelly to initiate trabecular formation. This is different from the zebrafish. In the zebrafish, my dear friend, Dr. Jiandong Liu, he found that is the cells can, the trabecular is initiated only by directed cell migration. Okay, so to test this hypothesis, one of the experiments we did here is we examined the cellular orientation here. So here's at about E, this is at about E7.5, about that stage. We can see the cells, this didn't display any orientation. But at about 26 somatic stage at the E9.25, you can see the cells here. The inner layer cells, those cells, the only the perpendicular to the heart surface, as you can see here. When we delete the NOM and NOM-like, we call it non-family proteins. In this knockout, you can see the cells, their orientation was disrupted. And we also use genetic model to show that the cells in the outer layer, they oriented parallel to the heart surface, while the cells in the inner layer, they oriented perpendicular to the heart surface. And here you can see this is a summary, this is a pie chart of the summary of the cell orientation pattern in the control. Each line here represents a cell, the orientation of a cell. As you can see, majority of the cells oriented perpendicular to the heart surface. And some cells only parallel to the heart surface. By the way, here it only means the cells in the inner surface, inner layer. While in the knockout, you can see majority of cells they align parallel to the heart surface. So you can see that the cells display orientation and the non-like are required for this orientation. And then we also want to see whether the cells can only go parallel and perpendicular orientation. So again, we use spindle orientation, mitotic spindles to determine the orientation here. As you can see, these dividing cells, their spindles are perpendicular oriented to the heart surface. While in these cells, the orientation is parallel. Then we use a genetic model to show that the cells, to determine the cellular orientation and also the cellular behavior during trabecular formation. So we use this confetti system here. So this confetti system is a very good tool to study single cell lineage tracing. So here's what we did here. So we using ROS26 Cre ERT2, which is inducible Cre, and ROS26 confetti, which is a reporter, has 4 different colors. And we induce Cre dependent recombination at about E7.75 with a very low dosage of tamoxifen. The dosage is so low that it only causes 1 or 2 cells to recombine in a single heart. And then we wait the embryos for 24 hours or 48 or 72 hours. And then we halve the embryos. And then we stain the embryos with PCAM and a DADI. And then we use chemicals to clear the embryo and image the whole heart. And then we analyze the clones. Okay, so here shows an example. That is, this is in a heart, we can see only 1 cell is labeled. Those are the cardiomyocytes. And here's endocardial cells. As you can see, in this heart, there's only 1 cell is labeled here. And I just show you an example that single cell can be labeled in the whole heart. And if we wait for 24 hours, we can image that cell can proliferate or migrate. So based on the geometry distribution of the daughter cells, we can infer what happened to the labeled mother cells. For example, here, you can see this labeled clone divided once, so they have 2 daughter cells here. And by the way, this is epicardial cells based on the orientation of the cells. And you can see the 2 daughter cells, 1 daughter cell still at the surface of the heart. The other daughter cells is migrated, is in the cardiac jelly. So based on this, we can infer that is the labeled mother cells, they divide perpendicularly. And so 1 daughter cell is keep at the surface of the heart. The other daughter cell is sent to the cardiac jelly. And in this scenario, you can see both the daughter cells are still at the surface of the heart. And then we can infer that is the labeled mother cell undergo parallel division. And so both daughter cells are still at the surface. What the third scenario, you can see that the daughter cells actually migrate into the cardiac jelly and will initiate trabecular formation. So we analyzed about 66 clones, and we found that 43% of the cells are derived, of the clones are derived from the perpendicular division, and 36% of them are derived from a parallel division, and 21% are derived from a directory migration. And what about the analyze the clones 72 hours after labeling? So you can imagine that is the cells, the labeled cells, if laid in vivo for 72 hours, that the labeled cells will proliferate multiple times and also will migrate to different locations. So based on the distribution of the labeled clones of those daughter cells from that labeled clones, we can infer what happened to the labeled mother cell, right? And here's just show you an example. That is, in this clone, you can see they have some cells at the surface of the myocardium and some cells, let's play it again. Some cells in the inner compact zone and some cells in the trabecular zone, right? So this scheme shows the distribution of the daughter cells. That is 1 cell or 2 cells still at the surface of the myocardium. And there are multiple cells in the inner compact zone and the multiple cells in the trabecular zone. So we analyzed 217 clones, and we found that those clones display 4 different patterns based on distribution of their daughter cells. One clone, one pattern is the surface pattern. That is, all daughter cells are still at the surface of the myocardium. We call this surface clone. And the second type, that is transmural clone, as I told you earlier, that is this clone, because they still have cells at the surface of the heart. And basically, it's transverse, because they have cells, distribute different layer of the myocardium, so we call it transmural clones. So this is the transmural clones. And the third type, we call it inner clones. sometimes, we call it trabecular clones because almost all the cells are in the trabecular zone. And the fourth type, we call it a mixed clone because they have cells at the surface of the heart and cells in the inner compact zone and also cells in the trabecular zone. So based on the distribution of these cells of the four type of clones, we can conclude that is transmural clones, inner clones, and the mixed clones contribute to the trabecular formation. Okay, so I want to tell you that is multiple genes, multiple pathways are involved in the regulation of trabecular formation and the trabecular morphogenesis. So as I told you earlier, that a signal interaction between the endocardium and the myocardium, such as NOTCH1, angiopoietin, unraveling, all those signal pathways regulates trabecular formation. And we found that that the NAM-LAC regulate this trabecular formation, and the integrin beta 1 also regulate this trabecular formation, and also N-cadherin regulates the trabecular formation, and we also found that the N-cadherin CDC42 regulates the trabecular formation. So this story is not published yet. Okay, so the third part, which is the one that I am going to focus on today, so that is beta-1 integrins regulate cellular behavior and the cardiomyocyte organization during ventricular wall formation. So this story was recently just accepted by cardiovascular research. Okay, so let me introduce integrin alpha, beta, and the delta ligands. So both alpha and beta are transmembrane proteins, and they have two alpha and beta subunit have two heterodimerides to form a receptor to bind the extracellular matrix and also interact with the active filament. So that's why integrin alpha beta receptors are important for the acting cytoskeleton. And actually, they're also involved in a signal interaction between the extracellular space and also intracellular gene transcription. And they have 8 beta subunits and 18 alpha subunits. And so beta-1 subunit can heterodimerize with 12 different alpha subunits. So each heterodimerization bind to different ligands. For example, beta-1 heterodimerize with alpha to bind to fibronectin, and beta-1 heterodimerize with alpha to bind to the ligands such as laminin-1 and laminin-8. So, and here lists the other combination of alpha and beta-1 and their ligands here. And so what about in the heart? We did a MR deep sequence and we found the expression transcription level of the different beta subunits and genes encode beta-1, beta subunits and alpha subunits. We found out gene encoding beta-1 subunit is expressed highest in the heart at about E10. And other beta subunits genes, regular beta subunits, including beta-5 and beta-8 and beta-3. While the alpha subunit that encodes encoded by alpha-6 and alpha-5 are the highest expressed in the heart. So that's why the life are interested in beta-1 and alpha-5 and alpha-6. So so let's talk about the data we have here. We found that beta-1 integrins and their ligands are asymmetrically localized in the myocardium. And this is a very busy slide. I wanted you to focus on here. So you can see the red here is integrin beta-1 staining. The green here is a cardiomyocyte marker. And based on those, you can see the integrin beta-1 is expressed in the epicardial cells here, and also in the endocardial cells here. And what I want to emphasize is integrin beta-1 is expressed in the cardiomyocytes. As you can see, if you pay attention to the expression pattern, they display an asymmetrical pattern indicated by the yellow arrowheads here. You can see this cells display asymmetric pattern that is enriched in the luminous side of the cardiomyocytes. And if we examine the distribution at the early stages, such as E8.75, you can see a different section of the heart here. All these cells indicated by the green arrowhead shows asymmetric distribution here. Again, they're enriched in the luminal side. And if we reconstruct this different sections here, you can see that it's beta 1 is enriched in the luminal side of the cardiomyocytes. And here shows a scheme demonstrating the asymmetric distribution. And here shows the quantification. That is, luminal side has a higher level of beta 1 integrin. Okay, you may ask, maybe the integrin beta 1 staining is not specific. Well, I want to tell you that the integrin beta 1 staining probably is the most clear staining I have ever had. That is because this is confirmed by the beta 1 knockout. So here's a beta 1, integrin beta 1 knockout. As you can see, the beta 1 stain in the cardiac muscle is completely gone. But in the endocardial cells, it's still there because NK715Q is not active in the endocardial cells here. So you can appreciate that is integrin beta 1 staining is very clear here. Okay, so actually this leads to the next question about integrin beta 1, their function in cardiac morphogenesis. So as I told you, we use NK715Q to delete integrin beta 1 in the myocardium. And then we call this knockout as a B1 knockout. So as you can see, B1 in the B1 knockout, integrin beta 1 deletion was complete. So the next question I'm asking is, as I told you earlier, beta 1 integrin has to heterodimerize with other alpha integrins to be functional. So we ask whether beta 1, what are the heterodimerized alpha units, and also what are ligands of beta 1. So we examine the first alpha subunit is alpha 6. So alpha 6, the localization pattern here, it's surprising to us in the beginning, because as you can see, alpha 6 is enriched in the trabecular cardiomyocytes here, and their expression in the compact zone is reduced. Okay, as you can see here, this is a higher magnification of zoomed picture. They are accumulated in the trabecular cells, but not in the compact cells. However, in the B1KO, you can see the localization of integrin alpha 6 is abolished. This actually suggests that alpha 6 only binds to beta 1 to be stabilized. Okay, and we also examine the ligands. So here is one of the examples, that is a fibronectin, which is a ligand for the integrin beta 1. Again, here's control. And surprisingly, that is fibronectin also display asymmetric distribution. That is, they are enriched in the luminous side of the cardiomyocytes here, indicates by the by the yellow arrowhead here. All the cells show asymmetric distribution to the luminous side. And then when we, by the way, I want to emphasize that is fibronectin is expressed in the cardiomyocytes here. However, in the B1KO, you can see the expression of fibronectin in the cardiomyocytes is almost completely abolished. However, in the inner compact zone here, I mean, in this layer here, in the basal layer here, you can see that they still have some fibronectin. Those might be secreted by the endocardial cells here. So I think here we can say that the fibronectin, it is a ligand for beta 1 integrins, and its stabilization requires beta 1 integrins. And we also examine other alpha subunits and other ligands, and we found alpha 5. By the way, so the function of integrin alpha 6 in the trabecular morphogenesis is being studied in the lab by one of the graduate students in the lab. And we also examine alpha 5. So the alpha 5, the distribution pattern is not surprising, and it is not reduced in the knockouts. And we also examine the ligands such as laminin 411 and collagen 4. So what I want to emphasize here is their expression pattern between the control and knockout. There's a much difference for both laminin 411 and a collagen 4. But both of them display a asymmetric distribution pattern. You can see they're accumulated to the luminal side of the myocardium for both the laminin and also for the collagen 4 here. So what it means to trabecular morphogenesis, we're going to talk more later in our model and the data later. And to continue to describe the phenotype of B1KO, so here we found that is B1KO has display outflow tract morphogenesis defect. That is in the control, you can see the dark lake. You can see the dark lake here display angle. The angle is about 70 degree. But in the knockout, you can see the angle actually is more than 100. This is actually suggesting there is a progenitor cell differentiation defect, and it usually will indicate the outflow tract morphogenesis defect. Okay, so let's talk about the second set of data. That is B1KO has display defects in trabecular formation, trabecular compact zone specification, and the cardiomyocytes organization. Okay, so let's talk about the trabeculation defect. So here shows a control. You can see the trabeculation indicated by the individual red arrows. But in the knockout, you can see the trabecular unit is missing, or the number is reduced. Here shows quantification. That is the number of trabecular per heart section was significantly reduced. However, if you check the compact zone thickness, you can see that the knockout thickness is much thicker compared to the control. Okay, and we also quantify the number of compact cardiomyocytes per ventricular section. You can see the knockout actually has much more cells than the control. And then we also quantify the trabecular cardiomyocytes per ventricular section. You can see the knockout has less. And here we use another way to quantify the cells in the trabecular zone and in the compact zone compared to congenital knockout. So what we found here is basically the cells in the knockout, they just fail to be properly organized to form trabecular. Okay. And we also examined the specification of the trabecular cardiomyocytes and the compact cardiomyocytes in the control and the knockout. And here we can see the H2 is expressed in the compact zone here. Okay, and it's weakly expressed in the trabecular zone here, and in the knockout, we didn't see there's a expression pattern difference because H2 is also expressed in the compact zone and it's not expressed in trabecular zone. However, the expression intensity in the knockout is reduced. Okay, and we examine the BMP10 here in the control is highly expressed in the trabecular cardiomyocytes and weakly expressed in the compact cardiomyocytes. And we examine that in the knockout, and we see different pattern here. That is, we can see BMP10 is highly expressed in both trabecular and the compact zone here. And here shows a quantification that is B1KO has high expression level and also different pattern from the control. Okay, so we then further examine what about the downstream of the BMP10 by staining the p-SMAD 158. So different from the control, we can see in the knockout, we can see a lot of p-SMAD staining in the cardiomyocyte, in the trabecular, in the compact, in both trabecular zone and the compact zone, which is is different from the control, suggesting that there's a trabecular zone compact zone specification defect for the as far as BMP10 as a marker, and which is further confirmed by the p-SMAD 158 staining. And we also use X3, which is a marker for trabecular cardiomyocytes. As you can see, it's enriched in the trabecular cardiomyocytes, and it's weakly expressed in compact cardiomyocytes. But in the knockout, you can see it's highly enriched in the trabecular in the compact cardiomyocyte, suggesting a pattern difference, a specification defect. And then we also stain P21. P21 is a marker for non-dividing cells, or it's a cell cycle inhibitor. And we can see it is expressed in the trabecular cardiomyocyte in the control. However, in the knockout, you can see it is also expressed in the trabecular cardiomyocytes. So this actually indicates that trabecular cardiomyocyte proliferation was reduced, and it also indicates a maturation relatively more mature than the control cardiomyocytes in the compact. And we use MPBA iron scope. MPBA again is marked for trabecular cardiomyocytes. It's enriched in the trabecular cardiomyocytes. But in the knockout, you can see they're expressed in both trabecular and both compact zone here. And so we also examine the actin filament, the sarcomeric array. Sarcomeric array, is an indicator of a cardiomyocyte maturation. So at an early stage, such as E9.5, the trabecular cardiomyocyte is more mature than the compact cardiomyocyte. This is evidenced by the actin filament and the sarcomeric array staining. As you can see, the trabecular cardiomyocyte has a good and a clear pattern of a sarcomeric array in the trabecular cardiomyocytes, but not in the compact cardiomyocytes. However, in the knockout, you can see they only form the actin filament and the sarcomeric array here, suggesting that the cardiomyocyte in the knockout in the compact zone is more mature than the cardiomyocyte in the compact zone in the control. And this is confirmed by the EM examination. Okay, the third set of data I want to discuss is the cell matrix, but not the encadenary cardiac jelly, apoptosis, and proliferation contribute to trabeculation defect and organization defects. Okay, so previously, we found that the encadenary in the mouse, and also in Didier Stanley's lab, they found encadenary in the zebrafish, is required for trabeculation. So we found that the encadenary distribution and by immunostaining and protein level by Western blot, we found that it is protein level and the distribution were not significant different. So we can fairly to say that encadenary distribution and protein level are not the factor that contribute to trabeculation and the organization defect in the B1KO. And we also examined the proliferation here, the cell death by staining the cleaved caspase 3. And then we found in the control and knockout only the outflow tract region shows cleaved caspase staining positive in the both control and knockout. And in the knockout, there's more in the outflow tract region. So this might explain why the outflow tract morphogenesis shows defects in the knockout. However, we didn't see any apoptosis in the atrial and in the ventricles, suggesting that the apoptosis is not a factor contributing to trabeculation defect and trabecular cardiomyocyte organization defect. And cardiac jelly is another essential factor that is required for trabecular morphogenesis and for compaction. So we examine if there's any difference for the cardiac jelly between control and knockout. So here stands a hyaluronic acid binding protein, or HABP, which is indicated for the cardiac jelly. And we can see, we didn't see a significant difference between the control and the knockout by immunostaining and also by Western blot. And we also examined the vesicle, which is a mark for cardiac jelly too. And by immunostaining and by Western blot, we didn't see any difference. And here shows the quantification. So we can say that the cardiac jelly is not a key factor for the trabeculation defect here and the organization defect in the B1KO. And we also examine cardiomyocyte proliferation between the control and knockout. So here we can see at E9.5, cardiomyocytes, proliferate more based on the BRD pulse labeling. As you can see in both compact zone and the trabecular zone, the cardiomyocyte proliferate at a smaller rate compared to the control. So it looks like that a proliferation might be a factor that contributes to the trabeculation defect in the knockout. However, we think probably this is not the case because E8.5 is a stage for trabecular initiation. And we found actually the cardiomyocyte proliferation, again, based on the BRD pulse labeling, we didn't see any difference there. So we think cardiomyocyte proliferation probably is not the key factor that contribute to our trabecular initiation defect in the B1KO knockout. Instead, we did an MR deep sequencing. We found the genes that regulate cell matrix and asymmetrical cell division and actual cellular space were changed, with significant change in the B1KO house. And those probably are the factors that contribute to the trabeculation defect. So we are further doing more experiments. So here, showing that the data sets showing that beta-1 integrins are required for cardiomyocyte orientation and organization. Okay, so here shows the cell size, cell orientation. So we measure the relative cell area based on the cell size, and we didn't see any significant difference between congenital and knockout. However, we measure the ratio of cell length to width, which is an indicator for cell orientation, okay, or for cell shape. And we found that their cell ratio, the ratio is significantly different. And we can see that is 72% of the cardiomyocytes, their ratio is larger than 1.5. While in the knockout, it's only 48% of the cells have a ratio that is larger than 1.5, suggesting that in the B1KO knockout, the cell orientation and the cell shape is a change. And we also measure the cell orientation. Measure the angles between the cell surface and the cell orientation. And here we measure the cells in the outer layer, the cardiomyocyte in the outer layer. And here shows the distribution pattern. We can see about 80% of cells, they display angle less than 15%. But in the knockout, it's only 34% of them display angle less than 15. And you can see some cells display a perpendicular orientation. And we also measure the cellular orientation for the cells in the inner layer. As you can see, that is the distribution pattern here. This is for the control. Majority of them, 40% of them display a perpendicular orientation. But in the knockout, you can see the perpendicular orientation percentage is reduced, suggesting that suggesting that the integrin beta-1 is required for the cellular orientation here. And we also use the EM to show that the cell orientation, the cell shape in the knockout is changed. Okay, the fifth set is integrin beta-1 integrins are required for cardiomyocyte onto cell division and asymmetrical cell division. Okay, well, first thing, I want to tell you that is the beta-1 is asymmetrically distributed in the asymmetrical cell division. As I told you earlier, that is integrin beta-1. It's enriched in the luminous side of cardiomyocytes, right? And what about in the asymmetrical cell division? So here's the asymmetrical cell division that is at the telophase. So you can see these two daughter cells, they actually are derived from same mother cells, because you can see the middle body of the two daughter cells are still linking the two daughter cells. So we can see these daughter cells has more integrins compared to these daughter cells. While in the parallel division, this is a parallel division. By the way, this is a perpendicular division because this orientation, the spin orientation is perpendicular to the hard surface. While this is a parallel division, we can see integrin beta-1 didn't show asymmetric distribution between the two daughter cells. And then we ask, what about when we delete integrin beta-1, where it affect the spin orientation here? This shows a perpendicular division again. This shows a parallel division. So we examine the spin orientation between the control and the knockout cells, heart actually. We can see in the control, 44% of them only a perpendicular division. But in the knockout, it's only 19% only a perpendicular division. And in the control, 25% only a parallel division. but in the knockout, it's 47%. This indicates that beta 1 integrins are involved in the spindle orientation of the cardiomyocytes. And we then ask, what about whether integrin beta 1 deletion disrupt the asymmetric cell division and disrupt the asymmetric distribution of self-determinants? So here we use HEY2 and BMP10. So for the HEY2 in the control cells, it's asymmetric distributed in the two daughter cells as the ratio is about 0.5 between the two daughter cells. However, in the knockout, the ratio is 1. This indicates that the HEY2 is equally distributed to the two daughter cells, and there's no asymmetric distribution in both perpendicular divisions and parallel divisions. And for the BMP10, the two daughter cells, the distribution of BMP10, the ratio is about 2. But in the knockout, it's 1, suggesting that there's no asymmetric distribution of BMP10 in the knockout. And this suggests that the integrin beta 1 is required for the asymmetric cell division, asymmetric distribution of self-determinants or determinants. This might explain why in the knockout there's a trabecular and a compact zone specification defect in the knockouts. Okay, and the data number set number 6. B1KO has displayed a reduced notch1 activation, but notch1 reduction was not sufficient to cause cardiomyocyte organization defects. So notch1, as I told you earlier, that is so critical. Beautiful data by the Jose's Jose's lab, showing that notch1 is required for trabeculation. So, and here we examined if notch1 activation was reduced. So what we found that is by immunostaining notch1 intracellular domain, which is the indicator for notch1 activation, we can see the number was reduced, percentage of NRCD positive cells is reduced in the knockout. And by Western blot, we can see total level of notch1 intracellular domain protein level is reduced. Here shows quantification again. And we also examined the readout for the unraveling pathway by staining phosphorylated ERP2. And we found that there's no difference based on the Western blot and quantification. So now the question is whether notch1 reduction is a key for the beta 1 knockout to display trabeculation defect and a cellular organization defect. To answer this question, to test this hypothesis, we use type 2 crete to delete notch1 in the knockout. So it's just consistent with previous results. That is, in the knockout, there's a trabeculation defect, but we still can see there's a trabecular. For example, there's a trabecular here, there's trabecular here, and there's a trabecular here, and then you can see all those indicated by the red arrows, shows there's a trabeculation. In the knockout, in the B1KO knockout, you can see their phenotype is much severe than the endocardial notch1 knockout. Suggesting deleting notch1 is not a key factor called trabeculation defect. What about the organization? And so we knock out notch1 and we examine the cellular orientation and the cellular size. And we found actually the cellular orientation and organization defect. We didn't see that. Here shows the orientation. We didn't see significant difference between congenital knockout. And also the cellular, the perpendicular and the parallel, we didn't see difference there. Suggesting that endocardial notch1 reduction in the B1KO knockout, it's not a cause. It's not a key factor for cellular organization defect in the B1KO knockout. Okay, so the last piece of data that I want to talk to is beta 1 integrins regular cellular behavior and organization autonomously. To answer this question, we use a very cool technique called a chimeric model. When I say chimeric, it means we delete integrin beta 1 only in some of the cardiomyocytes, and then we examine the defects of this integrin deleted cardiomyocytes. So if, for example, here, the two cells, which are labeled by confetti, Deutsche 23, we can see integrin beta 1 expression was abolished in these two cells, but not in the other cells, right? So just this is beta 1 integrin null cells, beta 1 integrin knockout cells. So we want to see the knockout cells. What about their migration, their proliferation, and their organization? And we found that in the knockout clones, their migration distance was reduced, which might explain this trabacylation defect. And also the cell number per clone was reduced, might explain why the cell proliferation was reduced. And we also examined the clone pattern. We found out in the control, the transmural clone is 44%, but in the knockout it's only 19%. And for the surface clone, in the control is 30, but in the knockout is 51%. So based on this piece of data, we can say integrin beta 1 regulates cellular behaviors and organization in a cell autonomous manner. Okay, so here's our conclusion. That is beta 1 integrins and the ligands are asymmetrically localized in the myocardium. And the beta 1 knockout hearts display defects in trabecular formation, trabecular compaction specification, and the cardiomyocytes organization. And beta 1 knockout cardiomyocytes display abnormal cellular organization. And beta 1 integrins are required for cardiomyocytes oriented cell division and asymmetric cell division. And beta 1 integrins regular cellular behavior and organization, autonomous. Here comes to our model. That is, we found integrin beta 1 that asymmetrically distributed in the cardiomyocytes, that enriched in the lumen side of the cardiomyocytes. And we found fibronectin and the collagen 4 and laminin actually that asymmetrically enriched in the luminous side. And the integrin beta 1 bind to the fibronectin to lock the cells in the scaffold or in the network, so the cells can display, can properly display orientation and migration and form the trabecular. When we delete integrin beta 1, then the cells fail to be locked in the ECM formed network or the scaffold, and the cells fail to be properly organized to display polarity and asymmetric cell division, and to be properly patterned to form trabecular. So this is our model. So I would like to thank the work is mainly done by Lian Jie, and with the help of other lab members too. And we also would like to, by the way, so I'll just list two lab members, former lab members here. We have much more, we have more members here. I didn't list here. So I also would like to thank Mark DePosen, who's our collaborator when I was at the Albany Medical College, and also with the help of Bob Schwartz at the University of Houston, and also Dr. E. Jen and Alan Burns and Arenas from the EM core at the UH and also UT Health. And this study was supported by R1 and AHA, postdoc fellowship and AHA SDG award. Thank you for your attention. I really appreciate that. Thank you. If you have questions, I'm more than happy to answer and discuss.
Wonderful. Thank you so much, Professor Wu.
Thank you.
I'm Abby again, and I want to open up the floor to see if anybody had any questions or comments about Dr. Wu's talk. Oh, and feel free to unmute yourselves or type your questions or comments into the chat box, and I'm happy to read it out for you. Any questions or comments? Thoughts? Yes.
Hi,
Yi-Chen
teacher.
Ding has one. Hi.
Hi, Dr. Wu. Can you hear me?
Yeah,
Yes,
sure.
we can hear you.
Yeah, perfect. So very nice talk. So, actually, I'm also working in cardiac imaging and also cardiac trabecylation field. So I'm very
Which
interested in
lab
your
you are?
talk.
Which
Yeah,
lab
actually,
you are?
you show a lot of very nice images. I have just two very quick questions. So the first one, when you talk about trabecylation in mouse heart, did you use a live imaging method, or maybe you only use immunostaining to label those specific cells?
Okay, depends on Well, we are not using zebrafish. Zebrafish, is nice. It's and can do the live image. For us, really depends on which methods. If it's for lineage tracing, then actually it's live because we label the cells and we let the cells go in vivo, and then we harvest them at the time we want. For example, 10 hours after labeling, or 24 hours after labeling, or 72 hours. So those are actually, although it's not live cells, but similar to live because everything's in vivo. And for the other staining, yes, we use fixed tissues to do the imaging. Yeah, it's different
I
from
see.
yeah. Do you
That
use
means
Please go ahead.
I see. So actually, that means when you are doing lineage tracing, actually that's like the in vivo data, right?
Yeah.
Oh, that's very, very nice. Probably I can talk to you more after the meeting. Yeah. So
Sure.
my second question actually relates to those kind of hypotheses in your trabeculation. You mentioned those kind of genes regulate trabeculation. Do you think hemodynamics actually contributes to those kind of trabeculation?
Oh, absolutely. I think both Didier Staining, Jen Liu, my friends, they proposed that that in zebrafish, I think in the mouse how too, just as I say, we these of some of the second pathway regulates this trabecular morphogenesis. But hemodynamics absolutely is one of the key essential pathway that regulates trabecular morphogenesis. And because the hemodynamics, will transduce signal from the endocardial cell to cardiac cells and the regular behavior of cardiomyocytes there. So I absolutely agree that that the hemodynamics is the key pathway. It's one of the key pathways regular trabecular morphogenesis there. Yeah.
I see. Perfect. Thank you for your answer.
Thank you.
Thank you.
Thanks for the question. Talk to you later.
Yeah, thank you.
Definitely, we'll help put you guys in touch. Anybody else have a question?
Hi, Mingfu. So Wase here.
Hey, Wase. So good to hear from you.
Yes.
I knew he was going to ask a question. Good.
Nice talk. So my question is regarding the single cell clones. Have you ever thought or you have done some special transcriptomics of these clones to find out, why they are behaving differently or how they are interacting with their surrounding cells?
Yeah, Wase, well, ideally we want to do that, right? But in reality, it's cost a lot. You have to do the single cell labeling and isolate the labeled cells from the cells that are not labeled by single cell sequencing, maybe a deep sequencing to see, their expression profile to see if they're different there or not. If you have the fund, if you have the energy, then it's good to try to do that. But we didn't do that. Was I think yeah, it's hard to answer this question.
All right, thank you. I have another question. So there's a
Sure.
recent published paper from I think it's Robertson lab, Robert Adamson lab. So that talks about the trabeculation, and they said that, the compaction thing is more to do with the slow proliferation of the trabeculi than the compaction. So what are your thoughts about it?
Okay, so we yes, one of the focus of the lab, is left ventricular non-compaction. I think what you said is this is one of the reason, yeah, one of the reason, and we have a different mechanism for that too, and we have different models to we establish the left ventricular non-compaction model to answer that question. So we think cell proliferation in the trabecular cells under certain situation contribute to the left ventricular non-compaction. We agree. But our study, using genetic reporters, linear tracing, and also drug treatment, chemical inhibition, chemical screening, we found, can be different, but we do not say that cardiomyocyte proliferation is not a factor. I don't know. I answer your question now. Was say.
Well, they argue with that normally what we have learned or what we know is like that, when the compaction process starts is that, the layer coalesce together. So in that paper, they have talked is more to do with the less proliferation of the trabeculi than the coalition, they do not coalesce together that much. So there is
Okay.
a big argument about, so that's a very recent paper actually.
Okay. Yeah, I think for compaction, they have two different mechanisms, okay? They have two different mechanisms. What you talk about is one of the mechanisms. There's another mechanism. So we call it, a malcompaction. He talked about a malcompaction. And the other mechanism is overtrabeculation. That's another mechanism. So yeah, so they are different. Different.
Okay, thank you.
You're welcome. It's good to catch up with you.
Thank you.
Wonderful. Well, I want to open up the floor again. But if there are no more questions, I'm happy to also wrap up and give a final thank you to Dr. Wu. We'll also send out a recorded link to the webinar with all the Q&A here at the end. And once again, thank you so much. And I hope that, congratulations, by the way, on your recent publication.
Thank you, Abby. Thank you for inviting me. It's real.
Thank you.
And also
Take care,
all the
everybody.
people to attend. Yeah, thank you. Bye-bye.